अस्सलाम वालेकुम स्टूडेंट्स। होप यू ऑल आर फाइन। स्टूडेंट्स, आज हम बायो क्लास 9 चैप्टर नंबर 9 एक्सरसाइज शॉर्ट क्वेश्चन आंसर्स सॉल्व करेंगे। सो लेट्स स्टार्ट। फर्स्ट क्वेश्चन है हमारे पास, डिफाइन क्लोनिंग। विच प्रोसेस ऑफ प्लांट is supposed to be a cloning? Answer is, cloning is the process of creating an exact genetic replica of an organism, cell, or DNA sequence. In plants, cloning can occur naturally or artificially through various methods. One common process of plant cloning is called vegetative propagation or asexual reproduction. This includes techniques such as stem cutting, cutting a stem section and growing a new plant from it. Next is leaf propagation, using leaves to grow new plants like with African violets. Next is division, separating a plant into sections, each with roots and growth nodes. Next is layering, bending a stem to the ground and allowing roots to form before cutting. Next is grafting, joining a stem scion to a root system rootstock. These methods produce genetically identical offspring, essentially clones of the parent plant. Artificial plant cloning can also be done through tissue culture, where plant cells are grown in a laboratory and then transferred to soil. Next question is, enlist any two suitable conditions for germination of seed. Here are two suitable conditions for seed germination. Number one is adequate water. Seeds need consistent moisture to germinate. The soil should be kept moist but not waterlogged, as excess water can cause seeds to rot. Next is optimal temperature. Seeds germinate best within a specific temperature range, usually between 20 to 30 degrees Celsius or 68 to 86 Fahrenheit. Some seeds, like those of warm season crops, require high temperatures, while others, like those of cool season crops, prefer lower temperature. Additional conditions that support seed germination include oxygen, light for some seeds, suitable soil pH, sufficient nutrients. Next is question number 3. Explain the structure of embryonic stem. Answer is, embryonic stem cells (ESCs) have a unique structure that enables them to differentiate into various cell types. Here's an overview of their structure. Cell shape. Embryonic stem cells are typically round or oval in shape with a large nucleus to cytoplasm ratio. Next is nucleus. The nucleus contains the genetic material, and embryonic stem cells have an open chromatin structure, allowing for easy access to transcription factors. Cytoplasm. The cytoplasm is minimal with few organelles, indicating a high nucleus to cytoplasm ratio. Cell membrane. The cell membrane is semi-permeable, allowing for the exchange of nutrients and waste. Adhesion molecules. Embryonic stem cells express adhesion molecules like ec-adherin, which help them adhere to each other, and the extracellular matrix. Next is surface antigens. Embryonic stem cells display specific surface antigens such as SSEA3, SSEA4, and TRA160, which distinguish them from other cell types. Next is telomeres. Embryonic stem cells have long telomeres indicating their ability to undergo numerous cell divisions. Next is epigenetic markers. Embryonic stem cells have specific epigenetic markers like histone modifications and DNA methylation patterns that regulate gene expression. This unique structure allows embryonic stem cells to maintain their pluripotency, self-renewal, and ability to differentiate into various cell types. Next is explain the role of livestock. Livestock plays a crucial role in various aspects of human life and the environment. Here are some of the key roles of livestock. First one is food source. Livestock provides meat, dairy products, eggs, and other essential nutrients for human consumption. Next is economic benefits. Livestock farming contributes to the economy through the sale of animals, animal products, and related services. Next is land management. Grazing livestock helps maintain open spaces, control weeds, and maintains ecosystem balance. Next is fertilizer and manure. Livestock manure serve as a natural fertilizer, enriching soil and promoting plant growth. Next is transportation and labor. In some areas, livestock provides transportation and labor for agricultural work. Next is companionship and recreation. Livestock like horses and donkeys offer companionship and recreational activities. Next is clothing and textiles. Livestock provides leather, wool, and other materials for clothing and textiles. Next is research and development. Livestock contributes to scientific research, improving human health and understanding of animal biology. Next is ecological balance. Livestock helps maintain ecosystem balance, influencing biodiversity and nutrient cycling. Next is cultural significance. Livestock holds cultural and symbolic value in many societies, representing tradition and heritage. Overall, livestock plays a vital role in supporting human well-being, economic development, and environmental sustainability. Next is question number 5. Explain the event of double fertilization in angiosperms. Answer is double fertilization is a unique event in angiosperms, flowering plants, that occurs during reproduction. It involves two separate fertilization events. First, fertilization. A pollen tube delivers two sperm cells to the embryo sac. One sperm cell fuses with egg cell oocyte to form a zygote which develops into the embryo. Second, fertilization. The second sperm cell fuses with the two polar nuclei or the secondary nucleus in the embryo sac, forming a diploid cell called the primary endosperm nucleus. This nucleus develops into the endosperm, a nutrient-rich tissue that supports embryo growth. Double fertilization results in a diploid embryo or diploid endosperm. This process ensures that the embryo receives the necessary nutrients for development and germination, while also allowing for efficient use of resources. Double fertilization is a key innovation that distinguishes angiosperms from other plant groups, enabling them to produce fruits and seeds that support diverse, diverse ecosystems. Next is question number 6. Explain the structure of embryo sac. The embryo sac, also known as female gametophyte, is a structure within the ovule of a plant that contains the egg cell and plays a crucial role in fertilization. Its typical structure consists of: first is egg cell or oocyte, the large central cell that will fuse with a sperm cell during fertilization. Next is synergate cells 2, two small cells that flank the egg cell, involved in guiding the pollen tube and facilitating fertilization. Next is two polar nuclei, two nuclei that fuse to form a secondary nucleus which will eventually merge with a sperm cell to form the endosperm. Next is antipodal cells 3 to 4, small cells located at the opposite end of the embryo sac from the egg cell, thought to be involved in nutrient uptake and support. Next is central cell, the large cell that contains the polar nuclei and will eventually develop into the endosperm. The embryo sac is typically 8 nucleate and 7 celled with the following arrangement: egg cell plus 2 synergid cells plus 2 polar nuclei plus 3 antipodal cells. This structure allows for double fertilization, which is unique to angiosperms and sets the stage for seed development. Next is question number 7. Justify that sexual reproduction in plants is more beneficial than asexual reproduction. Answer is sexual reproduction in plants is more beneficial than asexual reproduction for several reasons. First one is genetic diversity. Sexual reproduction involves the fusion of gametes from two parents, resulting in offspring with unique combination of genes. This increases genetic diversity, making populations more resilient to environmental changes and diseases. Next is adaptation. Genetic variation from sexual reproduction enables plants to adapt to changing environments such as shifting climate conditions or evolving pests. Next is hybrid vigor. Offspring from sexual reproduction often exhibit hybrid vigor, meaning they grow stronger and healthier than either parent. Next is disease resistance. Sexual reproduction allows plants to combine genes from different parents, reducing the likelihood of inherited diseases. Next is evolution. Sexual reproduction drives evolution, enabling plants to respond to selection pressures and evolve new traits. Increased fitness. Sexual reproduction can lead to increased fitness as offspring with advantageous traits are more likely to survive and reproduce. Asexual reproduction, on the other hand, produces genetically identical offspring, limiting genetic diversity and adaptability. However, asexual reproduction has its own advantages. such as rapid reproduction, colonies, preservation of desirable traits. Ultimately, both sexual and asexual reproduction have their own benefits, and plants employ a combination of both strategies to maximize their reproductive success. Next is define briefly apomixis, pollination, male gametophyte, apical germination, fertilization, binary fission, budding, sporangiophore, rootstock, bulb, rhizome. इन सबको हम वन बाय वन डिफाइन करेंगे. फर्स्ट ऑफ ऑल हमारे पास आ जाता है एपोमिक्सिस, a type of asexual reproduction where seeds form without fertilization, producing offspring genetically identical to the parent. Next is pollination, the transfer of pollen from the male reproductive organ anther to the female reproductive organ stigma of a plant, allowing fertilization to occur. Next is male gametophyte, the pollen grain which contains the male gametes, sperm cells necessary for fertilization. Next is apical germination, a type of seed germination where the seed coat remains above ground and the cotyledons, seed leaves merge and grow upward. Next is fertilization, the fusion of male and female gametes, sperm and egg cells to form a zygote resulting in sexual reproduction. Next is binary fission, a type of asexual reproduction where a cell divides into two identical daughter cells commonly seen in bacteria and protozoa. Next is budding, a type of asexual reproduction where a new individual grows from a bud or outgrowth on the parent organism, often seen in yeast and corals. Sporangiophore, a specialized structure that produces spores typically found in ferns and other cryptogams. Rootstock, the base of a plant including the roots and lower stem used for grafting onto a scion, stem and leaves. Next is bulb, a type of underground storage organ often consisting of layered leaves used by plants like onions and tulips to survive unfavorable conditions. Last one is rhizome, a type of underground stem that produces new shoots and roots, allowing plants like ginger and turmeric to spread and propagate. Next question is write the difference between asexual reproduction and sexual reproduction, artificial vegetative propagation and natural vegetative propagation, scion and rootstock, grafting and stem cutting, bulb and rhizome, binary and budding, hypogeal and apigeal germination, self and cross pollination. First one is difference between asexual and sexual reproduction. Asexual reproduction involves a single parent, produces genetically identical offspring, and can occur through various methods like budding, fragmentation, or vegetative propagation. Sexual reproduction involves two parents, produces genetically diverse offspring, and involves the fusion of gametes, sperm, and egg cells. Next is artificial vegetative propagation and natural vegetative propagation. First one is artificial vegetative propagation. Human-induced methods like stem cutting, grafting, or tissue culture to propagate plants. Natural vegetative propagation occurs without human intervention like runners, suckers, or rhizomes to produce new plants. Next is scion versus rootstock. Scion, the stem and leaves of a plant grafted onto a rootstock to produce a desired combination. Rootstock, the base of a plant including the roots and lower stem used for grafting a scion. Next is grafting versus stem cutting. Grafting, joining a scion to a rootstock using a specialized technique. Stem cutting, a method of vegetative propagation where a stem section is cut and grown into a new plant. Next is bulb versus rhizome. Bulb, a underground storage organ with layered leaves like onions or tulips. Rhizome, a underground stem that produces new shoots and roots like ginger or turmeric. Next is binary fission versus budding. Binary fission, a type of asexual reproduction where a cell divides into two identical daughter cells. Budding, a type of asexual reproduction where a new individual grows from a bud or outgrowth on the parent organism. Next is hypogeal versus apigeal germination. Hypogeal germination, seed germination where the seed coat remains below ground and the cotyledons grow downward. Germination, seed germination where the seed coat remains above ground and the cotyledons grow upward. Next is self-pollination versus cross-pollination. Self-pollination, pollination occurring within the same plant or between genetically identical plants. Cross-pollination, pollination occurring between genetically different plants, often resulting in increased genetic diversity. So students, हमारे ये क्वेश्चंस कंप्लीट हो चुके हैं. होपफुली आपको समझ आ गए होंगे. If you have any doubt, you can ask in comment section. Please like, share and subscribe my YouTube channel. Thank you.